Welcome to video 4 in this course on exploratory data analysis. And here we're talking about the theory or basics behind principal components analysis. This follows on what we talked about in the last part on dimension reduction, feature selection, and feature extraction. And that will lead on then when we talk about MDS, TS&E, and UMAP as well. So PCA is one of many approaches for transforming high-dimensional data where you have n dimensions into a more simpler form where we have k dimensions and that's more manageable. It's an unsupervised approach and therefore is very amenable to any kind of quality control or verification methods that might be used. I might think about, our questions as whether um, are we managing the data in the right way. So you might have variables within our data, such as age and height, that are very, very correlated, and then maybe allocated to the same principal component. These are separated from other ones that are uncorrelated and not dependent on these particular features. And what we typically are interested in is where we have a small number of principal components, and that this allows us to plot the data in two dimensions. So, for example, the PC1 is the biggest, most important principal component with the highest fraction of variation explained, and PC2 is the next biggest, and PC3, and so on through all the PCs. So this is a transformation method where we normalize the data in a matrix in the matrix X, and we look at the noise across the major axes of variation. So if you see across this axis, all the patterns are correlated there, and there's a separate axis of variation across this axis as well. And what we think about when we're looking at any particular set of data, a set of features, for each feature pair, if they're called X and Y, you might look at the variance of X, the variance of Y, and covariance of X and Y together to then obtain the correlation between X and Y. Now, if they're very correlated, they'll be allocated the same principal component. If they're anti-correlated, if they have a negative correlation coefficient, they won't be allocated the same principal component. So in detail, in more steps, we look at we get the mean of each column, and we get the mean of the columns and substitute uh, subtract that from the value that's shown, so that the average mean of across all the columns is now 0. So this is now normalized data. We then look at the covariance between each column pair. So if we've some heights in one column and some weights in the other column, we'll look at the covariance across all the different all data from going from I equals 1 to n that is present. So we'll take the difference between the height I minus the average height, and because this is normalized, this will be 0, and that the weight I from the weight 0 average, which is going to be 0 as well. And this will allow us to look at the correlation between the covariance between W and H. And then we'll be able to use the relative ratio of that covariance between W and H versus the variance of W and variance of H to extract the correlation coefficient. So we can say that if the covariance of height and weight is 0, then that they're independent. And we use our ratio of the correlation coefficient using this formula to then estimate how big that is. If we take the square of the value, we can say the heights and weights are of a small positive correlation, where 10% of one explains 10% of the other. It doesn't mean height causes weight or weight causes height. That's a separate question. So inferring causal links from that is not necessarily wise. From that, what we will do is get the eigenvalues of the different principal components that are present and the eigenvectors. The eigenvectors are in the new coordinates of the heights and weights, and they can be very, very different from the original ones. So they're not necessarily reflective of the true locations in a 2D space of the different features. And then what we'll look at PC1, and that's the axis with the largest number of variance explained across the principal components. Apologies, snips is put here, but that's not what we want. And PC2 is the second biggest, and PC3 is the third, and so on. So we can plot them like this, and we'll have some sort of level of um, measure for the variances here, which might vary a lot. But what we'll do is look at the fraction of variation explained across PC1 and PC2 and 3 and 4 and so on. And there's there are methods to determine how many PCs you should look at, but it depends what you want to get from it. But typically, the first four PCs will be most you would typically want be interested in. So this will allow us to see some different patterns of covariation. And one consideration is that we're assuming our data has linear effects and is a single peak that is unimodal. So it's like this. If our data has some violates these assumptions, we may get a horseshoe effects. So if we plot PC1 and PC2 and we scale the widths of the height and width of the plots by the um, sum of squares of all the PCs to get the fraction of variation explained, we can visualize it more effectively. And what we wouldn't want to see is this kind of nonlinear effect. So this is a horseshoe effect because it's shaped like a horseshoe, where we see the principal components are correlated in a peculiar fashion, yielding this horseshoe shape. So they're partially co covariating, but in a nonlinear space. And a solution to this is to use MDS or an equivalent method instead. So PCA is very good at many things, but it's not our only option here, and that's why we're going to look at other methods as well. So let's consider how we can create data to analyze, and what we're going to do in the demonstration videos later is look at microbiome data. And we're going to have three groups: healthy, IBD, and IBS. So irritable bowel disease, irritable bowel syndrome, and OTUs um, are taxonomic units um, that are kind of biologically unique. So they could represent taxa like a bacterial species. And we're going to look at this data across different groups. We're going to use RNorm to generate them and then bind together with RBind to create one big table. And we'll have a 40 by 40 matrix where we have 40 samples and 40 OTUs. And this will create a very large matrix of data using the matrix function. And what we'll try to make sure is that the matrix is has no negative values. Um, yeah. So what we'll look at using PCA is the number of principal cones in that data, and to see if they can be differentiated at the principal component level, to see if there's actually very strong, clear patterns that differentiate the different groups across the PC1, PC2, and so on. What's nice about PC1 is that as interpret PC all the PCs is that PCA provides interpretable axes, which means we can obtain the OTUs associated with a particular PC and make inferences about them, and that can tell us more about the taxa that differentiate, for example, PC1 versus PC2. Um, One consideration is that we want to uh, normalize our data during the PCA process, and that will try to mitigate this issue that very extreme values or large values can affect our outputs very strongly. So that's why we want to make sure that the data we put into it is accurate and visualize it so that we can verify that we don't have particular samples that are extreme outliers. Um, what we can consider as well that also that the PCA is not good for looking at raw compositional data, so it's the relative counts. It could be the relative counts of taxa, it could be the relative counts of cell types, it could be the relative count of ecological data. So we need to use a centered log ratio transform first. So how do we do PCA in R? We can use the prcomp function and many others. prcomp is my preference because we can extract information about the fraction of variation explained by a particular dimension very easily. There are many other ones and many other PCA methods. So for example, we have sparse PCA, negative and sparse PCA, non-negative iterative partially squared analysis, NIPALS as it's known, independent components analysis, kernel PCA, probabilistic PCA, neural network-based nonlinear PCA, consensus PCA, and many more. So there's many methods depending on the exact data that you're looking at. But if you're just trying to simply to plot your samples using PCA to do some quality control, it's very effective that, and you can use prcomp in base R to do that, and it'll do it very well. So that's the end of this introductory video. Please see the next one on MDS that will extend this further.